Hello all, welcome back to Invodent embryology sessions. Today we are all going to learn about a process known as spermatogenesis. Have you ever thought, why does males have a prolonged reproductive lifespan than compared to the females? Even in their older age group, males are able to produce their sperms. How is that possible? What are the reasons behind the origin of sperm cells? That is genesis, the origin of sperm cells. So that is known as spermatogenesis. Let us try to understand the process of spermatogenesis. How actually is it taking place and where is that happening? In the seminiferous tubules of the male reproductive system, the process of spermatogenesis takes place. Whenever we see the seminiferous tubules, we can witness two types of cells. One type is spermatogonial cells, the other type is serotoli cells. So serotoli cells are the nourishing cells, so take them apart. The other type of cells are spermatogonial cells. Spermatogonial cells are the mother cells for the production of the spermatozoa. So this type of spermatogonial cells with 44 autosomes and 2 sex chromosomes, these type spermatogonial cells are the mother cells to produce the spermatozoa. They undergo the mitotic division to give rise to two types of cells. Every spermatogonium cell will divide to give rise to two types of cells. Type A spermatogonial cells and type B spermatogonium cells. Type A spermatogonial cells again enter the cycle of spermatogenesis and act as a new mother cell to produce the spermatozoa. So that's why there is the production of the mother cell in the division of spermatogonium every time. So every time a spermatogonial cell divides, it will give rise to other mother cell which is capable of producing the spermatozoa. So the cycle repeats in the male reproductive systems, enabling them to produce the sperm cells even in their older age groups. So keeping that aside, discussing about the type B spermatogonial cells. Type B spermatogonial cells enlarge and divide mitotically to give rise to primary spermatocytes. These primary spermatocytes undergo first meiotic division. So as we have already learned in our previous sessions, the meiotic division, we will see the division of chromosomes and giving rise to two types of haploid cells. So there are two haploid cells that have been seen here because of the primary meiotic division. Then these primary spermatocytes, when divided, giving rise to two haploid set, again individually they will divide second meiotic division and give rise to two spermatids. So spermatids are the circular germ cells. They are circular in shape. They contain nucleus, centriole, Golgi apparatus, mitochondria, all the cellular components without a tail. So those doesn't have a tail. They are circular in shape. That's why they are known as spermatids, sperm-like, something sperm-like. So, and then they undergo the process of maturation and acquire a shape of a sperm. Each spermatid will give rise to one sperm cell. So this is how actually the sperm is being produced. So now the parts of the sperm cell we will be seeing here. So now the spermatid, as I have mentioned, the spermatid contains the Golgi apparatus. So the Golgi apparatus differentiates to give rise to acrosomal cap. So this is known as acrosomic cap, which is derived from the Golgi apparatus. And this is the head part of the sperm cell. This head part of the sperm cell is derived from the nucleus. And this is known as the middle part, wherein the middle sheet is covered by the mitochondrial cells. You can see the middle sheet is covered by these mitochondrial cells. And then there are centrioles. This is the proximal centriole and this is the distal centriole. The distal centriole will become a ring shape and it will form the annulus. And this is the axial filament what we can see here. So this is how the entire mature sperm cell is being formed. Now what happens is that initially when these sperm cells are produced in the seminiferous tubules, these sperm cells are actually immature and they are non-fertile. So they cannot fertilize an ovum. So when these are carried away through the epididymis, so there is a current fluid which comes in the seminiferous tubule. There will be a current fluid which carries these sperm cells through the epididymis. In this process, they acquire maturation and ability to move. They become motile. Further, the complete motility and the ability to fertilize is acquired during the ejaculation. What happens during ejaculation is that these sperm cells or spermatozoa get mixed up with the secretions of seminal vesicles and the prostate secretions. So they become fertile to fertilize an ovum. Then what happens? There comes the process of capacitation. The process of capacitation is something that the glycoprotein layer which covers the sperm cell will be altered. So when these sperm cells are entering into the ovum, so what is that ovum containing? Ovum is always protected by a circular structure, something like this. This is nothing but zona pellucida. So zona pellucida is always protecting the ovum. So how come this sperm cell pierces the zona pellucida and able to fertilize the ovum is that when the acrosomal cap comes in contact with the zona pellucida, there occurs some reactions which enable the production of lysosomal enzymes. So what are lysosomal enzymes? The enzymes which have the ability to digest the membranous structures. So these lysosomal enzymes help the sperm cell to penetrate through the zona pellucida and enter the ovum, and thus fertilizing it. So the changes taking place in the zona pellucida are known as zona reactions. So this is how the sperm cell is produced, matured and capacitated and getting fertilizing an ovum. So this is what is the process known as spermatogenesis. So now we have understood what is spermatogenesis. This topic is quite important in the exam point of view. From the examination point of view, when we look at spermatogenesis, the important things to learn is that about trisomy and monosomy. In our due course, we have listened many times this word of trisomy and monosomy. So what is actually happening in trisomy and monosomy is that during the formation of the gametes, the reproductive cells, there will be abnormal meiotic divisions that happens. So when there is an abnormal division, like when I have said that there are 46 chromosomes, so which should undergo meiosis and should give rise to 23 and 23 number of chromosomes haploid sets equally. So 46 divided by 2 will be having 23 and 23 in the healthy division. But what happens in abnormal meiotic divisions is that there will be one extra chromosome going into one daughter cell and one deficient chromosome which can be witnessed in the other daughter cells. So there will be abnormal meiotic division that is happening in this condition. So what happens if there is abnormal number of division of chromosomes? With this 24, they undergo the second meiotic division. There will be no reduction of chromosome number as usually. So there will be 24 and 24, 22 and 22 here. So one extra chromosome is always present in this type of division. What happens with this extra type of chromosome when it is fertilizing the normal cell, normal gamete with 23? So 24 plus 23 giving rise to 47. Actually, what should be the number here? 46. So there is one extra thing that is known as trisomy. So actually the extra chromosome is coming out. So this is causing trisomy. So depending upon the chromosome that has the trisomy, the disease will be established. If there is a trisomy of chromosome number 21, then it is Down syndrome. So if there is abnormal extra chromosome is coming, there will be reproduction will lead to the abnormal female to be developed. And if there is an extra Y chromosome, because of that, there will be the development of the abnormal male. So this is the abnormalities that we can witness because of the trisomy, extra chromosome. So what is happening here we will see. During the meiotic division, 24 trisomy fine, but there is deficiency. It has to be 23, but it is 22. So there is the deficiency of a chromosome. So this deficient chromosome further undergoes second meiotic division, 22 and 22. When this 22 chromosome will fertilize a normal gamete with 23, when these two undergo the fertilization, they will be obviously deficient of a chromosome. So this deficiency of the chromosome leads to monosomy. So this is 45. Actually the number should be 46. So a single chromosome is deficient here, resulting in monosomy. So these are the abnormalities and diseases resulting due to the abnormal meiotic divisions during the gamete productions. Hope you have enjoyed the session. Stay tuned to Invodent. Stay super motivated. Thank you so much.